So, hi Naren, are you there?
Yeah, yes, I am. Hold on. Can you see me?
So, very good afternoon, everyone. So, on behalf of the department, I welcome all of you to the second part of the lecture series, where today we have two eminent speakers. First one will be Dr. Naren Ramanan from Indian Institute of Science, Bangalore. And the second speaker will be Professor Suman Jain from AIIMS, New Delhi. Moving on with the first speaker. So let me introduce Naren to all of you. So Naren graduated with a PhD in molecular microbiology from the Institute of Molecular and Cell Biology, National University of Singapore. He then joined the laboratory of Professor David Ginty as a postdoctoral research fellow in the Department of Neuroscience, the Johns Hopkins University School of Medicine in Baltimore. After completing his postdoctoral training, Naren joined as assistant professor of neurobiology in the Department of Anatomy and Neurobiology, Washington University School of Medicine, United States. He moved his laboratory to the Center for Neuroscience, IISc Bangalore in 2013. Prestigious awards. He was a PDF awarded by Damon Runyon Cancer Research Foundation, fellowship from Whitehall Foundation, and the prestigious Swarna Jayanti Fellowship from Department of Science and Technology, Government of India. So presently, Naren's lab works on understanding astrocyte development and their role in neuroprotection in the CNS using a combination of mouse genetics, molecular, biochemical, and cell biological approaches. Over to Naren.
Thank you, Palak. Can you see my screen?
Yes, sir.
All right, perfect. So, one second. All right. Thanks, Palak, and thanks for this invitation. And I'm glad to share the work that's going on in my lab for quite a few years now. So as Palak mentioned, most of my lab is focusing on understanding how astrocytes develop in the brain, and most importantly, how these cells can provide neuroprotection in the brain following injuries, and also in several neurodegenerative diseases. Before we get into the data, I just want to introduce you to what neurodegenerative diseases are, in case you haven't heard about them. And I'm just going to focus on a few of them, and which would then become relevant to the talk that I'm going to be presenting on. So these diseases are in general very progressive, and the major hallmark that you would see for all of them is loss of neurons, which is irreversible, as these cells cannot be replaced. And the question is, what causes these? And the genetic mutations are varied, and very specifically and interestingly, in each of these neurodegenerative disease, you will see a particular population of neurons being very vulnerable to cell death. And the most common among them is Alzheimer's disease. And this is literally the poster child of neurodegenerative disorders caused by loss of neurons and synapses and memory and cognitive deficits. And mostly the neuronal loss is seen in the cortex and then slowly spreads to hippocampus. And of course, there are regions that are completely spared of neuronal loss. The hallmark of this disorder is the deposition of what's called as beta amyloid plaques, and also phosphorylated tau, the neurofilament protein, as neurofibrillary triangles. And for all of these diseases, I have to say there isn't currently any drugs available as cure. Anything is purely, and especially for Alzheimer's, after spending billions of dollars by various companies, there is currently no therapy available to help these patients. And same is true for several others. The Parkinson's is caused by a specific loss of dopaminergic neurons in a small region called the substantia nigra, pars compacta. And the molecular hallmark, again, is in patients is deposition of aggregates of this protein called alpha-synuclein as Lewy bodies and Lewy neurites. And phenotypically, the major issue is movement disorder because the output is control of dopamine, the striatal neurons important for motor control, which is what would happen when you lose these dopaminergic neurons in the substantia nigra. And you have the Huntington disease, and I would say this is the clinically the purest genetic disease where the cause is due to mutations in this gene called Huntington, which then leads to aggregation of mutant Huntington, and there's loss of striatal neurons, these GABAergic neurons, interneurons in the striatum, and the phenotype is loss of motor cognitive control and psychiatric disturbances as well. And not mentioned here, the other disorder is ALS, or so called the motor neuron disease, which is loss of motor neurons in the spinal cord and leading to loss of slowly motor control and leading to respiratory failure and death eventually. So as you have seen, each of these conditions have loss of particular kind of neurons. And so the work so far in the field has been concentrating particularly on the neuronal mechanisms involved in the death of these cells. Why do these neurons die? What do these mutations do to them, etc. But however, work in the last decade or so have started looking at the contribution of non-neuronal cells, and particularly the glial cells in the brain. There are two different classes of glial cells in the brain. These are called the macroglia, the oligodendrocytes, whose function is to myelinate the axons, providing faster neuronal transmission. And the other class of macroglia are the astrocytes, and which are the largest number of cells in the brain. By almost twice the number of astrocytes compared to neurons that you see in the brain. And the astrocytes in general perform a lot of important homeostatic functions in the brain. They provide nutrition, they help in removing excess ions from the environment, glutamate in particular, provide energy, also help in formation of synapses, removing of synapses, and also act as stem cells in the adult brain. So they do a lot of homeostatic functions. And one particular thing that stands out, especially when we talk about neurodegenerative disease, is their response to these injury or disease conditions. So this is what you would see in a normal brain that is stained for a marker for astrocytes called GFAP, which labels the intermediary filament protein. And so you see the structure of these astrocytes, of course, not the full structure, but enough to identify them. But when you have the brain that is under distress due to injury or neurodegeneration, these astrocytes undergo a plethora of changes which are morphological, transcriptional, and functionally different from the astrocytes in the healthy tissue. And this process is what we call as reactive astrogliosis, just gliosis. And these become much larger. And the major function of reactive astrocytes is, of course, to help in bringing homeostasis to the tissue that is under stress. And they help in actually removing tissue damage or limiting tissue damage, restore homeostasis. And it's can vary from either a milder response to a very severe response of gliosis. When you have injury, these astrocytes can actually form a glial scar that helps in limiting the damage from extending or expanding to the healthy parts of the tissue. And these astrocytes have also been shown to have very regionally specific response in terms of becoming active, reactive. But the main problem is when what starts off as being a protective response in the brain of these astrocytes to become reactive is when they actually become maladaptive. So when the injury is prolonged or in conditions of neurodegeneration, which is a progressive change in the architecture and function of neurons, these astrocytes that initially become beneficial may actually start contributing to the disorder itself. And work in the last decade or so have actually started to show that astrocytes can actually contribute to the progression of the neurodegenerative conditions. So what are the neuroprotective functions of astrocytes, of these reactive astrocytes? One is debris clearance. When you have neural injury, the astrocytic and help in removing the cellular debris. If the disease or the infection or injury that actually breaks the blood-brain barrier, these astrocytes help in actually sealing this blood-brain barrier again, which is a very important protective barrier for the brain. Like I said, if the injury is very severe, they form a physical barrier called the glial scar, and that helps in preventing the peripheral cells from entering into the healthy part of the brain, number one, and they can also help in preventing the secreted inflammatory cytokines from also spreading to the healthy parts of the brain. Inflammation. So they help in controlling the inflammation in the brain by producing anti-inflammatory factors and also suppressing the pro-inflammatory signals. Glutamate toxicity, which is one of the major hallmarks of neural injury and also quite a few of these neurodegenerative disorders, is excess glutamate being produced by these dead and dying neurons, and the astrocytes play a very important role in actually removing these glutamate. So there are very specific astrocyte glutamate receptors that actually help in this process. So these are the different ways that we know of where the astrocytes can actually help in restoring the homeostatic functions following an injury or also in neurodegenerative conditions. When we look at what do we know about the mechanisms that regulate, we know a few and there are a lot that we don't know. The astrocytes can become reactive in response to a lot of these changes. We have seen degenerating neurons as one a trigger that makes them reactive. You also have accumulation of these toxic proteins that can also trigger. But the major pathway that we know that actually is important for the astrocytes to become reactive is the cytokine pathway leading to activation of JAK-STAT pathway and the STAT3 as a major mediator of this conversion of these astrocytes from being a non-reactive to a reactive state. Mutation studies have shown that mice lacking this activation of STAT3 to a phosphorylated active form, or when you remove STAT3 itself, these astrocytes do not become reactive. And those studies were done to ask how important is the generation of reactive astrocytes for the tissue. And oftentimes they have seen that preventing these astrocytes from becoming reactive always tend to have a detrimental role to the tissue where healing was poor, blood-brain barrier doesn't get sealed, glial scars don't form. The other factor that is important for reactive astrocytes is this NF-kappa B pathway, and normally studies have shown activation of NF-kappa B in these reactive astrocytes is more correlated with a detrimental role for these astrocytes, providing a beneficial role to the body, to the tissues. There are also other studies that have shown that deletion of the FGF signaling or beta-1 integrin signaling or the sonic hedgehog pathway can lead an astrocyte to a reactive state in the absence of any extrinsic signals. Play very important intrinsic role in sort of acting as a brake on the normal astrocytes from becoming reactive. But of course, the question is, what is the nature of these astrocytes that are generated in the absence of these signaling pathways is largely unknown. And whether these astrocytes are good or whether these astrocytes are not good for normal neural functions. What sort of spread this work really well is the reactive astrocyte conversion has been known for decades, and it's a evolutionarily conserved process. In all conditions, people have shown that astrocytes do become reactive. But the question is, how different are these reactive astrocytes generated under each of these different conditions? And the first step towards understanding that came from the work from Ben Barris's group, when they actually took two different paradigms of injury. One is mimicking bacterial infection using lipopolysaccharide. These are small fragments of bacteria cell wall that actually induces inflammation. The other is a stroke model, a middle cerebral artery occlusion model of ischemic stroke. And what they did was they subjected the mice to both of these conditions, and then they isolated the astrocytes from the brain after a certain time, and then they looked at the gene expression profile of these astrocytes. And interestingly, when you stain them, they all look reactive, but when you look at the molecular profile, this study showed, the main study was Jennifer Semenian's in 2012, that actually showed that these two astrocytes, reactive astrocytes, have completely different patterns of gene expression. And from that, they showed subsequently that the astrocytes induced by neuroinflammatory signal, LPS, are actually neurotoxic because their expression profile indicated that they are not capable of supporting normal functions. And these astrocytes were then shown actually to secrete factors that kill neurons and oligodendrocytes in vitro. These astrocytes can no longer support normal synapse function and formation. They have decreased phagocytic activity, can actually contribute to neuronal death after exotomy. So this pathway of astrocytes induced by LPS is actually was called as neurotoxic, and they call it as A1 and A2. And this terminology is no longer used in the field because of a lot of other studies that came out showing astrocytes do not polarize into these exclusive states. The A2 astrocytes they found are the ones, or neuroprotective astrocytes, as they call, only based on gene expression profile, is induced following ischemic stroke. And these astrocytes seems to have upregulation of factors involved in neuroprotection, like neurotrophic factors, etc. But the question is, what are the mechanisms that actually regulate generation, not of A2, but astrocytes that would have more neuroprotective functions is the key. So, but if you look at the literature in the last 4 or 5 years since these papers came out, and people have actually associated the reactive state of the astrocytes to primarily a detrimental role in the brain, so much so that if you block reactive astrocyte generation in mouse models of Alzheimer's disease and the ALS, it actually is beneficial for the brain, suggesting that under conditions of neurodegeneration, the state of the reactive astrocyte is actually not good for the brain, and opening up two major questions that we sought. So what are the molecular mechanisms that actually can convert these astrocytes to a neuroprotective state? And if so, can these reactive astrocytes provide neuroprotection under different conditions as well, which is largely unknown in the field. And we sort of got interested or got into this when we were studying the role of a transcription factor called serum response factor, in short, SRF, in the brain and in actually its role in neural development and in glial development. This is a stimulus-dependent transcription factor, meaning that transcription factor gets activated in response to extracellular signals. It's very widely expressed in every cell in the body. And earlier studies have shown that if you delete SRF in the mouse at the very beginning, these mice are embryonic lethal around embryonic day 7 to 9 due to lack of mesoderm or heart formation, because SRF is very, very critical for cardiac development. SRF binds as a dimer to DNA, and actually, yeah, like I said, it's expressed in every cell in the body, but it actually provides tissue-specific functions by associating with a whole family of transcriptional co-activators that actually determines what SRF should regulate in a fibroblast versus in a cardiomyocyte versus in say, a liver or a lung cell. So to understand what SRF may do in the brain, so the work started off in my lab at WashU. My second graduate student, Paul, deleted SRF only in neural stem cells and used this with the Cre-LoxP technology, where we generated a LoxP floxed mouse and crossed it to a Cre recombinase enzyme that is produced from downstream of a nestin promoter. And this mouse was characterized before to show expression exclusively to the neural stem cells in the developing brain and nowhere else. So you would end up having a mouse when you cross these two, in which SRF is deleted only in neural stem cells. I'm not going to go into the details. What Paul found was SRF deletion in neural stem cells doesn't affect neurogenesis, but it has very specific effect on glial differentiation. Both production of astrocytes and the oligodendrocytes was largely affected in these mutant mice. So that sort of indicated that SRF might actually play a role in glial development and not in neurogenesis. So to sort of understand this further, we crossed the same SRF floxed mouse to another 3D combinase mouse, and in which case the 3D combinase is actually regulated by the GFAP promoter, which expresses exclusively in astrocytes. And in this mouse, a very, very productive collaboration over a decade now with David Gutman, deletes SRF only in astrocytes starting embryonic day 16 and a half, and the astrocyte starts to form. And we have characterized this line, and we show that the expression is very specific to astrocytes. We don't see expression of Cre in neurons in oligodendrocytes as far as we can tell. So what we did was these mice were bred, and then we looked at what happening to these astrocytes. And at 1 month of age, what we see is, and something that we also observe as shown by the other groups, GFAP gene gets downregulated in the cortical astrocytes after about 2 to 3 weeks of age. This is a transcriptional repression of the GFAP promoter, reasons that we don't know. So the GFAP expression is seen in the corpus callosal fibrous astrocytes and in the astrocytes in the hippocampus. To show that the astrocytes are there, we labeled with another marker, which is a pan-astrocytic marker, a better one than GFAP, which is ALD H1L1, alcohol dehydrogenase, that you can now see the astrocytes in the cortex. If you look at the mutants at a month of age, what we see is this massive upregulation of GFAP signal and also ALD signal, both in the cortex and in the hippocampus and also in the white matter astrocytes. So, like I said, the GFAP in a large population of astrocytes in the brain gets downregulated in the adulthood. And the only time that the expression comes back on is when the astrocytes are becoming reactive. And in response to injury or infection, etc. But the fact that we are seeing this upregulation of GFP in a normal, healthy brain otherwise indicated that these astrocytes were actually becoming reactive when we delete SRF. And we further showed that this reactive astrocytes are present in every part of the brain that we looked at, in the neocortex, in the hippocampus, striatum, callosum, cerebellum, etc. And we can actually see that these astrocytes are also larger in size. Another phenotypic hallmark of astrocytes when they become reactive is that they become hypertrophic, much, much larger in size. And which is what we see. You can see these astrocytes are barely discernible in a control brain, but you can actually see them because they are much, much larger in size. So this indicated that SRF deletion in astrocytes causes them to become reactive, and it's not restricted to any particular region in the brain. The gene the pathways, the FGF, the sonic hedgehog, beta-1 integrin that I introduced to, actually those papers have shown that deletion of these in the brain, in astrocytes, actually only causes reactive astrocytes in a very region-specific manner where these pathways are more active than the other. But contrary to those in SRF mutant mice, we actually see a widespread a conversion of astrocytes to a reactive state. We further confirmed this by labeling for other markers of reactive state, which is vimentin, another intermediary filament protein, nestin as well. And what we see is, compared to the control, we see upregulation of these genes in the mutant mice, indicating that, in fact, these astrocytes have become reactive once SRF is deleted. The next question we asked is this a transient response that we see at very early stage, or this is a response that actually continues? And what we see, even at a year of age, these astrocytes continue to be hypertrophic and reactive in every part of the brain that we looked at compared to the legitimate controls. But interestingly, these astrocytes do not seem to affect any aspect of neuronal development, because the neuronal numbers, both at 5 weeks of age and 1 year of age, we don't see any change in neuronal numbers in all the regions that we looked at, indicating for the first time to us that these reactive astrocytes are likely not neurotoxic as those shown for those produced by LPS induction. But of course, the question that what we have seen so far, SRF is necessary for neural stem cell to astrocyte differentiation. When we delete SRF in developing astrocytes, we see a reactive phenotype that is persistent and widespread. However, these reactive astrocytes do not seem to affect neuronal survival in numbers throughout the brain, indicating that they are probably not toxic. But the problem is when you delete in a developing system, a response that you get could very well be because of a perturbation to the developmental process, right? So the question is, the reason why we see reactive astrocytes is it because of some other perturbation to the astrocyte differentiation that is going on, or are we actually stumbling upon a break that converts these astrocytes to a reactive phenotype? Or in short, is SRF required in a cell-intrinsic manner to regulate this conversion from a non-reactive state to a reactive state? To address that, we actually turn to, and in the first mouse that we use, the deletion happens very, very early when the astrocytes are beginning to form. To actually see if SRF is necessary for normal astrocytes to stay in a non-reactive state, we crossed our mouse, our SRF lost mouse to a third mutant mouse. And in this mouse, you also have the CRE enzyme regulated by the GFAP promoter so that it's astrocyte specific, except there is a modification to the CRE recombinase where it is actually tethered to the ligand binding domain of the estrogen receptor. So when you do that, when the CRE is made, the estrogen receptor will keep it engaged in the cytoplasm along with the heat shock protein 90. And this estrogen receptor ligand binding domain is actually mutated so that this doesn't bind endogenous estrogen, but instead it actually binds to an analog of estrogen called tamoxifen. So now you have a mouse in which CRE will be made in astrocytes, but it will stay in the cytoplasm until you can choose the temporal window in which you want to delete SRF or any gene of interest. And when you do that by injecting tamoxifen, then tamoxifen will bind to this ERT2 site, and due to the conformational change, the heat shock protein gets kicked out, and now the pre-tamoxifen bound ER complex moves to the nucleus and then causes deletion or recombination between the LOXP sites. So now this gives us a very, very strict temporal time window to delete SRF, and that's exactly what we did. So what we did was to take these mice and inject tamoxifen or the carrier oil as a control at 2 months of age. We know that the astrocyte development in all respects is complete by around 4 to 6 weeks of age in terms of its maturation of structure, etc. So by 2 months, we can be certain that the development of the astrocytes is largely complete. So we now inject tamoxifen at 2 months of age, and then we looked at the astrocytes and what becomes of them at various time points from the start of tamoxifen injection. So we first looked at 2 months of age. In a control mice or an oil-injected knockouts, this is what we see: barely no GFAP expression and faint GFAP expression in the hippocampal astrocytes. But when you look at the SRF-ER mutant mice, we now start to see hypertrophic astrocyte with very robust GFAP expression, indicating that these astrocytes have become reactive. So this suggested that SRF actually plays a cell-intrinsic role, and it is what we saw that the straight knockout line is not a developmental defect, but rather indicating an intrinsic role played by SRF to regulate its conversion from a non-reactive state to a reactive state. We looked at later ages at 12 months, and these astrocytes, in fact, persist as reactive astrocytes throughout the life of the animal. We have looked at mice that are even 24, 26 months of age, and what you trigger at 2 months of age stays the same until the mouse dies. We also confirmed that with other markers. In fact, they are indeed reactive in their phenotype. Then we looked at microglia because the other the microglia perform phagocytic function and immunological functions. These are equivalent to the macrophages in the brain. And these also respond to changes in the brain and can become reactive and can actually perform both beneficial and detrimental roles. So we wanted to know whether microglia are affected in these mice. And when we looked at 2 months, when we first see reactive astrocytes, we don't see any microglial activation when we look at IBA1 expression levels. And also the number of microglia also do not change both in the control and the knockout mice. But when we looked at 12 months post-injection, now we see a robust upregulation of IBA1, indicating that the astrocytes have become reactive. And we also see an increase in cell numbers, indicating that the microglia have expanded in response to the astrocytes becoming reactive. And what we know compared to 2 months and 12 months, it's the reactive astrocytes that are converting or causing this microglia to become reactive as well. So the next question we wanted to know, given the role of reactive astrocytes normally seen in neurodegenerative condition, which is not supportive of a lot of neuronal functions, is to ask whether the SRF deficient reactive astrocytes, are they beneficial or are they detrimental to the normal mouse brain? So first we looked at synapse numbers. We stained the hippocampus and cortex for pre and post synaptic markers, colo and UA1, and counted the number of synapses. And what we found, both at 3 months post-tamoxifen injection and in much older mice, 15 months post-tamoxifen injection, no significant difference in the number of pre- and postsynaptic co-localized puncta, indicating that the synapse numbers are not affected in the brains of these mice. We looked at LTP, and this was a very, very fruitful collaboration with James, who you would have heard yesterday. So James Chalaya and his graduate student Vijaya helped in doing some of these field recordings from the mutant control mice hippocampal sections. So we looked at 3 months post-tamoxifen injection. We don't see any difference in basal synaptic properties of these control and knockout mice. And when we looked at LTP, which is sort of a measure, physiological measure of learning and memory, we don't see any significant difference in the mutants compared to the control. And we also find very similar at 15 months post-injection, that's 17 months of age. We don't see a significant change in basal synaptic properties, definitely no difference in LTP between control and mutant mice, indicating that the presence of this persistent presence of reactive astrocytes is not detrimental to normal neuronal activity and normal neuronal functions. We also did behavior, and we found that there is no significant difference when we looked at hippocampal dependent spatial learning. This is a context conditioning, contextual fear conditioning that requires the hippocampus, wherein you put the mouse from its home cage into a particular space and you train it to associate that space with a shock. And once the mouse gets trained, if you put this mouse back into that cage and you look at the time that it takes for the mice to freeze, the faster it freezes, it means that the mouse immediately knows that if I'm in the space, I'm going to get a shock. And when we do that, and then you compare that with context 1 and context 2, because if it goes to context 2, which is a space that is not associating with shock, the mouse doesn't freeze as much compared to context 1, where it is trained to actually get a shock. We see no significant difference between control and mutants, indicating that the knockouts have learned to associate that space with the shock, and they haven't forgotten that. The other measure of spatial memory is Barnes maze, where the mouse is learned. This is a maze, as you can see, with holes in this plate, and there's one hole which will have the escape door for the mouse to actually get into a box. And you have external cues for the mouse to know where is, or which quadrant, or where exactly is this hole for it to escape. And when you do that, you train these mice, and then you, after the training is done, you see how fast it takes for the mouse to get to that hole. Or when you don't have the escape box, you actually see how much time it spends in a quadrant where the hole was supposed to be there with the escape entry. What we found was no significant difference between the control and knockouts, the latency to escape, how long it takes to reach the hole, distance, how fast it goes in terms of distance, and the time that it spends in a quadrant where the hole had the escape entry point. We find no significant difference between the control and knockouts, indicating having these reactive astrocytes in the brain doesn't seem to affect behavior as well. Next, we wanted to know what is the molecular nature of these astrocytes. So we actually isolated astrocytes from control and mutant mice, and we ran RNA-seq experiments. And very interestingly, what we found was the genes involved in defense response, cytokine production, defense to virus, and defense to bacterial infection, and type 1 interferon signaling pathways were highly upregulated in the mutant astrocytes compared to the control. Nothing that we saw this indicated that these astrocytes were neurotoxic to the brain, because none of the pathways that we see have been associated with functions that are detrimental to the brain. We validated the changes in these genes by real-time PCR. So we do see several of these interferon regulated genes upregulated. We also see a lot of these chemokine and cytokines being activated in the brain. But interestingly, these astrocytes do not seem to upregulate the pro-inflammatory cytokines, interleukin-6, TNF-alpha, and INOS, and which has been shown to be the genes mostly upregulated in astrocytes that are reactive and tend to have a detrimental role in the brain. So having seen that these astrocytes are persistent, don't seem to affect any of the normal functions of the brain as we could test, and the fact that they seem to be upregulating genes involved in general for protection in terms of innate immunity and viral and bacterial defense, we wanted to ask, are they actually reactive, actually protective, or detrimental to the brain? So we looked at two different categories of injury and disease. We looked at brain injury models, three of them: traumatic brain injury, a neuronal excitotoxicity model, and a stroke model. And we started off with two most common neurodegenerative disorders, Alzheimer's and Parkinson's. And we asked whether these mutant mice with reactive astrocytes are actually better compared to their control under these conditions. And the first is a cortical stab wound injury. This work was carried out by, it's being continued by Surya, a very talented postdoc. Very similar, a very simple model. What we basically do is expose the brain, and you inject a Hamilton syringe or a scalpel into the brain. And after a sufficient period of time, you look at the closure of the wound, the healing of the wound, how fast it takes, and so on and so forth. And this is what we do, expose, and this is exactly, we make a very tiny window in the skull, and then we insert the Hamilton needle as a model for penetrative traumatic injury. And after 10 days, we euthanize the mice, and then we look at the area, volume of the wound, etc., to get a sense of whether the astrocytes are going to help in the recovery process or actually going to be prolonging the recovery process. This is what we get in a control. You can actually see where the needle had gone through. And this is a top-down view when you take horizontal sections. This is what we see at 10 days of age. And what you see, this intense GFAP staining around the hole is where you would see actually the glial scar that is showing that staining. When we looked at the mutant mice, what we see is much smaller wound cavity area. And as you can see here, it's much smaller compared to the control at 10 days of age. And looking at the area of the wound, it's much smaller, indicating that the astrocytes, these wounds are actually healing faster than the control does. I have to point out that Surya is completely blind to the genotype of these mice, so he doesn't know the mice that he's handling is either control or knockouts until after all the staining experiments are done and once the measurements are taken. But it becomes quite evident when you actually put GFAP, because you can easily see which is the control and which is the mutant mice. The second model we used is excitotoxicity, and this is a more widely used model. Canic acid is an analog of glutamate, and it's 30 times more potent in causing neuronal death than glutamate is. And excitotoxicity is one of the major drivers of neuronal cell death following ischemic stroke, and in a lot of neurodegenerative conditions as well, so much so that if you can help these astrocytes in removing glutamate, you can actually see lesser neuronal death in those models. So what we did for this is basically we did intracerebroventricular injection of canic acid. We did do a stereotaxic injection into the ventricles here so that we can have very nice effect on the hippocampus. So the mice are anesthetized and canic acid is injected into the ventricle. And then after the mice come out of anesthesia, about an hour or so, these mice go into tonic-clonic seizures, which we measure to tell how severe the seizures are, and indicating that the canic acid is in fact working. And after about an hour of seizures, these mice usually recover. And we then, after a week, we analyze them, the brain, and particularly the hippocampus, for cell death by two different approaches. So this is what we see in the control and knockouts. The canic acid injection, day 7, the mice are euthanized. We label them with pseudo-JHC, which labels all dead neurons and degenerating neurites, axons and dendrites. And you can see it in the hippocampus from rostral to caudal axis. You see massive cell death as expected in the control. Surprisingly, it's what you see, and surprisingly, we see very negligible cell death in the mutant mice compared to the control. And this is quantified here in terms of pseudo-JHC positive cell numbers. When we looked at tunnel and other means of looking at dead cells, we see exactly the same. We see massive cell death in all the 3 regions of the hippocampus, whereas in the mutant mice, we see very little to negligible cell death in response to canic acid, which is very surprising to us, indicating that these reactive astrocytes are actually able to remove glutamate very efficiently, and probably that's one way that they could be providing neuroprotection under these conditions. For stroke, we used a different model, also very widely used, an MCAO, the middle cerebral artery occlusion model, where you block a particular branch of the cerebral artery, is one model that is used, but another one, which both of these have pros and cons, where this one is more defined. You can choose which artery you want to block in a much more defined manner than you would with MCAO. And the stroke is not as larger in this compared to MCAO because it's a very small region that you actually occlude. And the way we do this is use the property of the dye called rose Bengal dye. And this is injected IP into the mouse, and after about 10 to 15 minutes, if you activate this dye by emitting a cold green light laser at around 532 nanometers, this causes photoactivation of the rose Bengal dye. And this activation produces singlet oxygen species that would then lead to platelet aggregation, endothelial damage, causing clot in a very small parts, resulting in blockade of blood flow and causing neuronal death. And so what we do is, if you are very careful in picking your region, you can pick the motor cortex, a particular branch that you want, block it so much you can actually get a phenotypic response, motor paralysis in that particular, the opposite side that you can measure. So we haven't gone that far yet, but we just started off doing this. So we injected rose Bengal, controlled knockout mice, then induced this photothrombotic stroke. And this is what you see in the control, very similar. You can see this is Nissl stained brain after, I want to say, 24 or 48 hours after the stroke induction. You can actually see from a rostral caudal plane the place where you had stroke induced, and this is the infarct. And this is the ischemic core and the ischemic penumbra. You can see virtually no cells present there. If you look at the knockout mice, what you see, as you can see here, is the infarct size was much smaller. And in fact, we do see a lot more neurons in the ischemic core compared to what we see in the control mice, and this is quantified here. This suggested to us that the mutant mice with these reactive astrocytes are able to protect the brain from stroke-induced neuronal death. This is the slide that I showed earlier. If you take the neurodegenerative diseases, the work in the last 3 to 4 years in particular have shown that the conversion of astrocytes under disease conditions, ALS, Parkinson's, Alzheimer's, is typically neurotoxic or a maladaptive state of these astrocytes. So much so that if you block this conversion, people have found that you can actually help in reducing some of the phenotypic changes in these mice models, including behavioral as well. So this showed that the state of these astrocytes, reactive astrocytes in a neurodegenerative disorder, is actually not good for normal progression of the disease. So that brings the big question for us: are these SRF deficient astrocytes? What is their state in conditions of neurodegeneration, and whether they're actually protective or not? To do that, we crossed our mice to a AD mouse. This is the APP Swedish mutation and Brasilin-1 deleted mouse. So we obtained these triple transgenic mice, and we stained them first for amyloid plaques. You can see stained for a particular amyloid-specific marker. You see very high deposits in the neocortex and in the hippocampus. And when you look at these triple transgenic mice, I'll explain what these two are. We actually see lesser plaques in the AD transgenic mice in which we have deleted SRF only in the astrocytes, generating these reactive astrocytes. We see very, very little plaques to almost no plaques in the other line. And this is quantified here. The difference between these two lines, and this suggested that the SRF deficient reactive astrocytes cause significant plaque reduction in the mouse model of AD. The difference between these two lines is in the first line, this is the tamoxifen inducible line, in which we gave tamoxifen at 2 months of age, and then we analyzed for plaques at 9 to 12 months of age, having reactive astrocytes for a time of about 5 to 8 months, and you see about more than 50% reduction in the number of in the amount of plaque. But when we also cross this with the other line, in which we have the developmental deletion of SRF along with the AD mutation, what we see is near complete removal of plaques, because in this mouse, the plaques were around for nearly a year, suggesting that longer presence of these astrocytes, or maybe earlier generation of reactive astrocytes, can actually prevent the plaques from either forming or accumulating. But we don't know whether these astrocytes are affecting plaque formation or helping in faster clearance, something that we're currently looking into. So, but this is promising to us that these reactive astrocytes are in fact protective. Lastly, we looked at the Parkinson's disease model. We used the 6-hydroxydopamine model, in which we injected 6-hydroxydopamine into the SNPC area directly. And this is a unilateral injection. We injected to one side, and 10 days later, we look at loss of dopaminergic neurons. And staining for tyrosine hydroxylase, a specific marker for these dopaminergic neurons in the SNPC. And this is what we see. This is the SNPC region, and you actually see there is a reduction in the number of neurons as you go from a ventral to caudal plane. And what you see in the mutant mice is significantly more neurons. And in fact, we don't see any significant loss of neurons when we compare the ipsilateral side, the side in which we injected 6-hydroxydopamine, to the contralateral side, which is the uninjected side. So if you look at these two purple with the ipsi and contra, we don't see anything significant in terms of neuronal loss, indicating that these astrocytes are able to protect these neurons from 6-hydroxydopamine-induced cell death, suggesting that they are indeed protective in a disease model as well. We're expanding this to the genetic model of alpha-synuclein, and also looking at another pathway of an MDTP induction. MDTP is a drug that if you inject IP can cause bilateral loss of dopaminergic neurons. So we are actually expanding this to ask whether the neuroprotection is seen in all the 3 models of Parkinson's disease. So what we have seen so far, this will be the concluding slide. We have shown that we have seen that SRF deletion in adult astrocytes also causes these astrocytes to become reactive, and they persist throughout the brain. They do not seem to affect or cause cell death of neurons, and we have also looked at oligodendrocytes, no death of oligodendrocytes. They do not affect synapse numbers doesn't affect basal synaptic transmission, LTP, learning and memory. They seem to show an upregulation of innate immune response genes. And in models of both injury and neurodegeneration, they seem to be providing a very broad neuroprotective function. And our work shows for the very first time that you could actually manipulate a genetic pathway to convert astrocytes to a broadly neuroprotective state. And we're continuing and finding out the mechanisms. We have a clue as to what SRF regulates to make them reactive. We're also data mining our RNA sequencing data to look for pathways that are actually providing neuroprotection under each of these different conditions. Finally, thanks to a lot of people without whom this work wouldn't have advanced so much so fast. Paul started the earlier work in generating these mutant mice and characterizing their initial phenotypes. Monica, a graduate student, currently a postdoc at ICANN, did the characterization of these mice and showing that that they become reactive. Surya helped with all the functional assays in terms of showing that they are indeed neuroprotective. Srividya, Sumitra, Priya helped along the way in a lot of generating of clones, RT-PCR, etc. And students that helped in other parts of the project. My collaborators within the department, Deepak Nair, Balaji Swanand, and graduate student Arnab that did the learning and memory experiments. Collaborators at JNC, James and Vijaya, funding from the our scientific institutions, DST, DBT, and Sefik Prakrat, and my collaborators at WashU in St. Louis. Thank you very much, and I'll be happy to take your questions.
Thank you, Naren, for the wonderful talk. A big round of applause for Naren, please. So Naren, can you take a few questions?
Yes, please, yes.
So the floor is open for the questions. Please go ahead.
Yeah, thank you, Naren, for this wonderful talk. First, I would like to ask you about this microglial cell activation, which was followed by the astroglial cell activation. Actually, when you have seen that there is a reactive astrocytes, there was a microglial cell activation in the aged mice. But when you saw it in the adult mice, like at the age of 3 months, there was no microglial cell activation. So what was the reasoning that like age dependently there was a variation in the microglial cell activation after the reactive astrocytes?
Yeah, good question. So I just made it simpler by showing 12 months. So we actually wanted to see whether the microglia become reactive, and if so, at what age. And so what we found is it takes about a month to I would say somewhere between 4 and 8 weeks for the astrocytes to become reactive once we delete SRF. And the microglial activation is not seen at that time at 2 months post-injection when we first looked at. The earliest of microglial activation is seen a month after the astrocytes become reactive, which is 3 months post-tamoxifen injection. And the microglial activation also then suss out the life of the animal. So I showed 12, so it's not an aging-dependent phenotype. We see even in the younger mice, a month after the astrocytes become reactive, or somewhere between 1 to 4 weeks after the astrocytes become reactive, that the microglia also become reactive. And that is known here and there in the literature. Typically, the microglia are the ones that first respond to these changes in the brain, produce cytokines that would then activate the astrocytes to become reactive. But in our case, since this is a cell-intrinsic phenomenon, it looks like and we see a lot of these chemokines upregulated by these astrocytes, at least at the gene expression level. So, and that's probably how they are activating the microglia. Having said that, we also have done an RNA-seq for the microglia in the mouse, and we actually find very similar upregulation of these innate immune response pathways in the microglia than anything that is associated with being detrimental. So that's why we don't see any ill effects of having microglial activation as well.
So you feel that it was a protective type of microglia, M2 phenotype?
Yes. But we haven't dwelled into it because of lack of resources and time. But we strongly believe that the microglia are actually in a beneficial state and not in a detrimental state. Yes. So they could be M2, but I don't want to use M1, M2 terminology there because that's also being taken away in the field because they don't seem to polarize into 2 states. But yes, we think that the microglia are not the bad guys as well.
Okay, thank you. Thank you.
So I have a question in the chat box, Naren. So the question is, the SRF knockdown increases gliosis. It's interesting that it does not affect the neuronal numbers. So it will be interesting to perform a traumatic injury where glial cells can cause scarring, and this scarring can hinder neuronal communication.
Yeah, so we
yeah.
haven't expanded on that because of the problems in expanding our colony for a lot of these experiments. So for each of these, the TBI, and especially we are actually looking at more details of scarring, in particular changes in connectivity, because as you would know, glial scars has been shown to be detrimental for regrowth of axons, axon regeneration. So we want to ask actually whether these astrocytes or scar that they form is actually in some sense beneficial for axonal regrowth. So we want to do both, either that and also a CST axotomy to actually ask CST crush to show whether there is effective regeneration in our mice models. So yeah, these are all sort of lined up as experiments to do. And an easier one would be the optic nerve crush to see whether that would be better regenerative in the presence of these astrocytes. Because when you induce the other class of LPS-induced astrocytes, an axotomy actually leads to increased cell loss and then being beneficial. So for each of them, yeah, we are now slowly expanding to actually micro observe all the changes that is happening from the time that we injure. So we want to do a more time scale, a temporal longitudinal study to actually see what are all the cellular changes happening. Plus, if we can tie it up with behavior, that would be great to actually see any positive cellular change is actually correlated with better behavior as well.
Great. So Naren, thank you very much for your precious time for all our students and participants who are online over here. And I wish if you could personally come over to NIPER-Ahmedabad in future so that we can host you and we have interaction with all our faculty and students in future.
Absolutely, absolutely.
Thank you very much.
Yeah. Thank you, everybody. Thanks, everybody.
So now I invite Professor Kherna to introduce the next speaker, who is an excellent speaker, and Dr. Kherna is going to introduce her.
All of you. Thank you for listening to the first lecture, which was very interesting, and I am sure that the second one will also be very much interesting lecture. So it will be given by Dr. Suman Jain, and I just would like to briefly explain her CV. So Dr. Suman Jain is professor at Department of Physiology, All India Institute of Medical Sciences, New Delhi, India. And Dr. Jain is a neurophysiologist, and her areas of interest and expertise are synaptic plasticity and neurodegeneration. Dr. Jain did her graduation in human biology from AIIMS, Delhi. And Dr. Jain has been actively involved in deciphering the translational potential of repetitive transcranial magnetic stimulation and the use of nanoparticles in patients, as well as animal models of spinal cord injury, Parkinson's disease, Alzheimer's disease, cerebral palsy, and other neurological disorders. Her lab has expertise in numerous electrophysiological, behavioral, biochemical, and morphological techniques. Besides these, Dr. Jain is also involved in understanding the effect of prenatal environmental enrichment on functional development of various brain areas. Dr. Jain is also a recipient of AIIMS Research Excellence Award and has a number of publications in peer-reviewed national and international journals. Dr. Jain is also a reviewer of many reputed international and national neuroscience journals and into review committees of various funding agencies. Currently, Dr. Jain is Vice President of Indian Academy of Neurosciences and member of IUPHR Committee of INSA. So today, Dr. Suman Jain will be presenting or giving a talk on neuromodulation of brain by repetitive magnetic field stimulation in neurodegenerative disorders. So thank you, Dr. Suman Jain, for accepting this invitation. And now over to you, Dr. Suman Jain.
Thank you, Dr. Karin, and thank you, Dr. Pallab, for excellently organizing this expert lecture series and inviting me to deliver a lecture. That's an honor to me. Can you please see my slides, and am I audible?
Yes, ma'am. Yes, ma'am.
So today we'll be talking about basically about repetitive magnetic field stimulation. So what is repetitive magnetic field stimulation is basically a non-invasive method of brain stimulation techniques. So either you can stimulate or modulate the brain, and it's a non-invasive technique. You may not put any electrodes, and it is quite painless. And this technique was introduced by Anthony Barker in 1985. So when he introduced basically, so there is a coil here which you need to put on the skull, and then you give a stimulus. So say if you're stimulating the motor cortex, you will get a motor evoked potential. So initially when he started, this was the non-invasive method of stimulating motor cortex to record motor evoked potentials and therefore to calculate the conduction velocities in under various neurological conditions. But later on, Pascal Leon and number of other scientists, they introduced the term as repetitive TMS, and which was then used or been under trials for various therapeutic and its various therapeutic potentials has been tested. So basically the principle on which this magnetic field works is electromagnetic induction. So what is electromagnetic induction? From our physics, we know that it was given by Michael Faraday, that if you're passing electric current through a coil, okay, through a wire or through a coil, then magnetic fields are generating. And if magnetic fields are changing, or if there's a changing magnetic field, that can also induce an electric current. And then how much current will be generated, that is dependent upon the number of turns in the coil, the area of the coil, and rate of change of the magnetic field, magnetic flux. So that is the phenomenon on which it is working, wherein this coil is placed on the skull. So this basically I'm talking of humans, and that is why I'm giving this principle basically more related to humans. Similar coils are also available for rats, which I'll be talking about. So what you do that in this coil, you pass the current, and therefore the magnetic fields are generated. Now the advantage of using these magnetic fields are that they can easily penetrate through the skull, bony skull, and then enter into the neural tissue. And within the neural tissue, then they can induce the electric current. Okay. Now, so these eddy currents or electric currents, therefore, since the neurons are excitable structures, so they can alter the membrane properties of these neurons are the glial cells, and therefore, either producing depolarization or hyperpolarization, and therefore changing the excitability of the neurons. So that is how these electromagnetic waves are working, or stimulation is being provided. So that is how it is modulating the neuronal activity, and therefore depending upon what condition you are using, we can either depolarize or hyperpolarize the neurons, right? So this is what it is being shown here, that so there's a TMS coil, the magnetic field is produced, and then if there's a rate of change of magnetic field will induce electric current in the tissues that will alter the membrane properties. But just to remember that in the cortex now, if neuron properties are being altered, then obviously we also have to know that neurons, all the neurons, they're not oriented in the same direction. So some could be, if you look at the TMS coil which is placed on the skull, some could be making an angle of 90 degrees, the other neurons could be making an angle of 0 or maybe parallel to the TMS coil. Okay. So it was proposed by Fox et al. In 2004 that how much current, effective current, will be induced in these neurons is actually dependent on this angle, which is a cosine angle. So cosine theta, as we call it. Okay. So if the cosine angle, if it is 90, then cosine 90 is 0. So minimum current will be induced in these neurons which are perpendicular to the surface of the skull. Whereas if the angle is cosine 0, then it is 1. Okay. And therefore maximum current will be induced, or maximum modulation of these neurons will take place. And in between neurons, depending on their orientation and therefore the cosine angle, the current will be induced. So that is the working principle of this TMS. So that is how it is affecting various neurons which are placed in various orientations. Okay. Other aspect of this is that when you're using a coil, then you are basically, even with the finest coils which are available in the market, the area which you are stimulating is slightly larger. So maybe it is not actually stimulating one single pyramidal neuron or a group of pyramidal neurons. Rather, it is covering a larger area. So that is, you can say that it could be a disadvantage of using TMS coil, that it is not directly stimulating the group of pyramidal neurons. Rather, so there is a more larger activation, and therefore, if you're recording from corticospinal tracts, we do say that actually it results in generation of indirect waves. So this indirect waves is due to the stimulation of larger area, and therefore there is a transsynaptic activation of neurons, of the pyramidal neurons in the motor cortex, and then they can stimulate the muscles. Okay, so it's an indirect activation through the trisynaptic connections that it results in activation of the facilitation of the stimulation of the corticospinal tracts. Okay. So there are various types of coils which are available in the market. So more commonly now is used as this figure of 8 coil. Okay. So in this figure of 8 coil, the current is flowing in such a way in the two coils that the maximum amount of current is generated in the neural tissue, which is underlying underneath the between the junction of these two coils. Okay. So therefore, the area which is being stimulated is smaller as compared to if you're using a circular coil. And this is a double cone coil, which is used for giving more deeper brain stimulations. Then this is H coil, which is a more recent addition to the coils, and this can also be used for deeper structures penetration, more than approximately like 7 centimeter, 6 to 7 centimeter. And then now we have available in the market the rat coil also. So we have now recently last year we have also purchased this rat coil, and we are now using this coil to do further experiments on animal models. Okay. When we started this work on magnetic field stimulation, so around 2000, 2001, 2002, we had started this work, that how the magnetic fields are affecting and what is the mechanism by which they must be bringing about their effects. So that time the coils were not available and it wasn't easier to fabricate a coil. So we made a magnetic field chamber, which was basically a modified version of the Lee Whiting coil. And it had these 4 circular coils, okay, 2 outer coils and 2 inner coils, and the current was passed. And if you can appreciate the central platform here where the rats are kept. So according to this, on this central platform, you will get a uniform magnetic field of 17.96 microtesla. So this we have indigenously designed, and we had fabricated this and done a lot of experiments and using this coil till quite late. Okay. So what I'll be showing experiments is basically by using this magnetic field chamber, where you can keep number of rats together also. But obviously they have to be restrained and kept on the platform in a perspex made chamber. Okay. So basically, therefore, the aim of the studies which we conducted using this magnetic field chamber was that to study the effect of magnetic field exposure on the morphological and functional changes in ICV-induced streptozotocin model. So Alzheimer disease, as previous speaker Nareen was also talking about, is basically a progressive degenerative disorder. So we have to remember, I am emphasizing on the words progressive. So it's a chronic disorder and progressive because it is gradually progressing, and it's a degenerative disorder, so affecting your neurons and therefore resulting in loss of memory. It's a very highly prevalent and second most, I think, the neurological disorder, which is highly prevalent in India and all across the globe. And it is expected that by 2030, maybe the number of cases will double. And therefore, and since the person survives the disease for maybe 15, 20, 30 years, so it is very huge financial burden to society and may get worse. And that is why it is very important that we should be able to do something with the Alzheimer disease patients, provide them some relief, and reduce this financial burden of the society. If you look at the therapeutic interventions for Alzheimer's disease, yes, there are a number of FDA-approved drugs, which are basically acetylcholine inhibitors or NMDA antagonists. And there are a number of drugs which are in clinical trials. So anti-inflammatory or antioxidant, metal chelator, or some even rehabilitation therapies. But what we are focusing, and more recently in recent years it has come up is magnetic field stimulation. So if you do, while I was doing the review of literature and I entered this in PubMed, then what you can see that Alzheimer's disease in reference to TMS, it is only in approximately in the last 20 years that we had number of papers coming up and more interest was generated that how the TMS is working, whether it is effective or not, and what is the mechanism of action. So still it is the TMS, lot of clinical trials are being done, even in institute, even at AIMS, TMS has been used, or some trials are going on in Alzheimer's and Parkinson's disease patients. Even in my lab, we are doing on Parkinson's disease patients. But what exactly is happening if you're giving this type of stimulation, and especially if you're giving the stimulations for longer duration, then what is the mechanism of action is not clear. So therefore, what as a physiologist or as a neuroscientist, we thought that we should be able to see that what is the exact mechanism of this magnetic field stimulation in Alzheimer's disease model. So all of you must be aware there are different types of models for Alzheimer's disease. Some are spontaneous, transgenic. That's what I think Dr. Naren was showing, ablation, and then chemical models. So we chose streptozotocin. Okay. So why did we choose streptozotocin? Because streptozotocin, as we know, is taken up by a glucose transporter, that is GLUT2, and then once in the neurons or even in the glial cells, then it can produce lot of effects. So there is a reduction in the uptake of the glucose, energy imbalance, excitotoxicity, mitochondrial dysfunction, and through GSK pathway, actually in streptozotocin, you can see A beta formation and HP tau, which are the hallmarks of Alzheimer's disease. So that is, the significance of using streptozotocin, that once you inject streptozotocin, it takes approximately 15 days for the cognitive deficits to occur. Okay, so that is one, that it is progressively damaging the neurons, and then after approximately 15 days, you find the cognitive deficits. And if it continues, then it has been shown that approximately after around 30 days, there is a formation. You are able to see neurofibrillary tangles, and then it takes another 45 or 60 days for A beta plaques to be formed. So therefore, if you have to compare with the human condition, then we found that streptozotocin is producing lot of deficits which are quite similar to the humans. So it does cause spatial memory deficit. It causes A beta deposition, neurofibrillary tangles, reduction in energy intake, mitochondrial dysfunction, oxidative stress, neuroinflammation, gliosis, and so on and so forth. Okay. So maybe some emotional components or visual spatial ability may not be getting manifested, but at least the hallmarks of Alzheimer's disease as seen in human patients can be seen if you're injecting streptozotocin. Mind you, this is intracerebroventricular injections. Okay, so this is not that you're injecting into the hippocampus. This is when you're injecting intracerebroventricularly the streptozotocin, then it causes all these responses appear gradually over a period of time. Okay, and so for this study, then what we did, we had control healthy animals, and then we had sham, means that we had just injected artificial CSF into the by ICV route, and then we had also a group in wherein we used the sham animals, and then we exposed to MF. So we wanted to see that if the Alzheimer's disease is not there, whether MF has any effect. So that group we had, and then we had AD group, and then AD plus MF group. Okay, so then immediately 24 hours after the surgery, we exposed them to this 17.96 very low microtesla for 30 days, and in some of the studies we had also done for 60 days. So most of the results today, which I'll be showing you, will be more focused on 30 days, and some of them will be for 60 days. Okay. So then we did a lot of behavioral studies and other morphological studies. So Morris-Water maze, as we know, is again for spatial memory. This is one of the standardized procedures which is being used to assess the spatial memory. So this is the Morris-Water maze. You must be aware of it. So there is a platform zone. Okay. So generally, if you leave the animals into water, okay, they can swim, but then they're afraid of drowning, and therefore they are in a lookout or explore that if there is anything onto which they can climb and will be more safe. And therefore there is this platform zone is there. So as they explore, they find the platform and they can climb on it. So gradually, so what we did, we gave 4 trials every day for 4 days, and then 5th day we removed the platform, and then we observed that how the animals are moving. So whether they're directly reaching to the platform zone or what is their exploratory nature. So all this was recorded by using the camera. Okay. And mind you that again, these behavioral studies were done in an isolated room. So the rats did not have any disturbance from outside or from even the student who was using it. And then all this was recorded. So we have Nolder's EthoVision system software, wherein we could get live recording of all these live tracking of the rats which are there. So this diagram shows the live tracking of the animal as well as the heat maps of the tracks. So what you can see this red dot basically here, which you can see is the platform in this 4th quadrant. Okay. So, and this is the visual cue, which I think forgot to mention. So there is a visual cue also. So the animal learns that it has to go to that zone. Okay. So this is the control animal. You can see it is moving around, but then more number of lines you can see near this, the platform. Okay. This was the sham animal. Again, you can see it was more towards the platform. Now, in AD animal, what you can see, so this is the platform zone. So it was just moving in the periphery. So it showed the thigmotoxic behavior, as we call it. Okay. Now, in sham plus MF, there was again more concentrated on near the platform zone. And even in AD plus MF, although the exploration was much more as compared to control or sham animals, but then they were more targeted towards the platform as compared to the other groups. Okay. So now coming on to the graph. So in the first graph, what we wanted to see that what is the effect on training, on the learning, that whether the animals, whether they were AD or they were exposed to magnetic field stimulations, whether they were able to learn this task or not. So this is showing you the escape latency to the platform zone. Okay. So you can see, as you can see, this triangle, purple color triangle, this is AD group. Okay. So in AD group, you can see there was a learning, and gradually over the days, they were able to learn that where is the platform zone. Okay. And similar to other groups, they were able to learn. Okay. However, in day 1, this learning took time. Okay. But definitely they were able to learn. Okay. Now, if you compare the baseline data with the post-surgery data, okay. So what you can see, this is again the AD group. There was a significant increase in the latency. So latency increase means they took more time to reach to the platform zone. Okay. And this was significant as compared to their baseline data. Okay. And then this C, D, and E, they are showing basically the different parameters and the comparison amongst the groups. Okay. So the first one is control, sham, AD, sham plus MF, and AD plus MF. Okay. So there was a significant increase in the escape latency to the goal quadrant. Okay. And however, in the MF group, though there was an increase when compared to the sham group or the control group, but there was, when compared to the AD group, then there was a significant decrease. Okay, right? And if you talk of the time spent, that how much time they spent in the goal quadrant, then we found that the AD rats, they were more, that's what I showed here, that they were more moving round and round. So they kept moving round and they did not spend much time in the goal quadrant. Whereas the MF exposed groups, they did spend a lot of time in the goal quadrant. Okay, and they stayed there. Okay, and this is showing you the escape latency to the platform zone, which is again the similar results that AD latent animals took longer time as compared to the other groups, and in MF there was a recovery. Okay. Now, then we also tested their avoidance memory. So we did the passive avoidance test, which was step down, basically step down type of test. So wherein, so we had done this test in a Skinner box, so which has a grid floor, and then there is a platform here onto which the animal is being left. So on day 1, basically, when the rat is being left on the platform, So the animal will obviously, it has an exploratory nature, so it will try to get down, and as soon as it get down, we give them shock. So that is day 1. Then day 2, that is 24 hours thereafter, we again leave the rat onto the platform, and then we see how much time it takes to get down. Okay, so if the rat remembers that there was a shock when it gets down, then it won't get down. Okay, however, if there is a memory deficit, then it should get down. Okay, so that is the basis of this passive avoidance task. So again, as you can see, this is the baseline. Okay, so all the groups, they had the similar type of latencies, but after surgery on 30 days, the latency to the step down decreased significantly in AD group. Okay, so that showing that there was a memory deficit in these rats, and when they were left on the platform, they immediately got down. Now, when you see AD plus MF group, definitely as compared to the other groups, there was a decrease, but there was a significant improvement as compared to the AD group. Okay, so this is again when comparison was done amongst the, this was basically comparison with the 30th day, but this is showing you the comparison amongst the groups. So you can see that AD, there was a significant decrease, and there was a recovery with when magnetic field exposure was given. Okay, and we did also counted number of errors. So number of errors which was made by AD group was significantly more as compared to the rest of the groups. Then we also tested their recognition memory. So for this recognition memory, we did novel object recognition task. So in this novel object recognition task, this is an open field arena. We generally keep in the first trial, we keep 2 objects which are similar to each other, and then the animal is explored to explore for 5 minutes, and then after delay of 15 minutes, then one object is replaced by another object. Okay, and so now the animal has to recognize that this object is different. And as you know, the rats, they explore more the novel object as compared to the familiar objects. Okay, and then a recognition index is calculated, that how much time it spent exploring the novel object as compared to the total exploration time, right? So again, these are the heat maps, okay, of the 2 objects, but I think I'll directly move on to the results. So this is the baseline data. Okay, after 30th day, as you can see, in AD group, the recognition index was significantly reduced, indicating that the animals, the rats, were not able to recognize the novel objects, and they were spending more or less equal or more time with the familiar. So they were more or less exploring both the novel and familiar objects. Okay, now if you do comparison amongst the groups, so you can see this is again the AD group. There was a significant decrease in recognition index. However, a significant improvement was seen in MF group. So in the AD rats which were exposed to MF, there was a significant increase, similar with the discrimination index. Then finally, last but not the least, we also did elevated plasmase. I think all of you must be familiar with it. So this is an elevated plasmase which has 2 open arms and 2 closed arms, and this is kept at a height of around 50 centimeters. And then we see that how the animal is moving, how many times it has gone to open arm or the closed arm. Okay. And this was basically to test the anxiety behavior of the rats after induction of AD model. And what you can see that the time spent in ratio of time spent in closed versus open arm was quite high in AD rats as compared to the other groups. Okay, and which was significantly decreased after MF exposure. Okay, and if you look at the head dipping durations, that is how many times they're dipping their head in the open arms. So that is again indicative of their anxiety behavior. So the AD animals, the head dippings were very less as compared to the rest of other groups, showing high anxiety levels in the AD rats. So having done the behavioral studies wherein we saw that there was a deficit in spatial memory, recognition memory, avoidance memory, and as well as the anxiety was quite high. So we did the CV staining in CA1 and C3 groups. So this is a control and sham groups. So AD group, we didn't find any neurons, much neurons, and as well as there was a thinning of the layer, okay, which in AD plus MF group, we did find some neurons here and there, okay, so they were sort of more widely distributed, and the layer was slightly more thick. Similarly, in CA3 layer, if you can see, there is a laminar organization of so-called CA3 layer. However, in AD group, this was totally disrupted and there was a neuronal loss. And in AD plus MF group, this was trying to recover. After that, we also did, we looked at the shapes of different type of the neurons in the different layers. So this is the typical shape of a neuron, we know about it. However, in AD group, you can find that the neurons were shrinked and they were in the stage of dying, and number of neurons, there was a significant neuron loss. However, after MF exposure, the neurons were, we did find more viable neurons, and they had a shape which was coming, which was quite similar to the original one, similar one with the CA3 pyramidal cells. Then we also did Golgi-Cox staining. So in Golgi-Cox staining, we looked at both the basal dendrites as well as the apical dendrites. Okay, and then you can see here that Golgi-Cox staining was done. So in the control animals, this is the typical neurons, and you can see the extensive profuse dendritic branching in these neurons. Okay, so this is a sham animal. Whereas in AD group, you can see there was a significant loss in both basal and apical dendrites. Okay, and you can see the shrinkage of the neuron was also can be seen very clearly in this group. Okay, and then this is sham, and then AD plus MF group, you can see that although then there was some sort of shrinkage, but it is trying to revive its structure, and you can see more of dendrites which are being present there in the CA1 layer. We did do for the other layers also, but I'm just showing you for one of the layers that how it was being done. Okay. So besides that, then we also, like I was saying, that in streptozotocin induced rats, we can see neurofibrillary tangles as well as A beta plaques. So this is Congo rat staining we did for A beta plaques, and so this is the enlarged version which of the AD group, because we were able to see the A beta plaques in the AD group. Now this was done at 60 days. So although I've shown previously a lot of data from 30 days, but this was specifically for 60 days we had done rats, because according to the literature in streptozotocin induced rats, we could find the formation of A beta plaques, or we can observe A beta plaques only after approximately more than 45 days. So we kept the animals for 60 days, and then we could see these, congophilic particles which were present along with some small nuclear cells. And this was not only present in hippocampus. This we did see in other areas also, like dentate gyrus, and there were some congophilic particles which also seen in cortex, frontal cortex also. So there was an extensive damage which was seen in AD animals, and deposition of plaque as compared to the rest of the groups. So, and then this is your HP tau expression, which was done at day 30 and day 60. Like I was saying, that again, literature says that we can see in streptozotocin induced STZ animal models, we can see HP tau after 30 days or at 30 days. So we did at both at 30 days and 60 days. Okay, so this is the AD group which you can see, and then AD plus MF group. So this is showing you at day 30. This is we did at CA1 area and as well as in CA3 area. So we found when we did the counting, so we found that there was a significant increase in AD group as compared to the MF group. Okay, so this is at day 30. This is at day 60. Okay, so in day 60 the increase was much more, and there was a significant decrease in MF group. And similar results we found in C3 area. So this is in AD group, and this is AD plus MF group. Okay, and then when we compared the 30-day group with the 60th day group, then we found that again, as the time progresses, there was an increase in the number. So there was a significant increase in the HP tau in this tangles between the two groups. Though in MF group also there was an increase in both the groups. Okay. So, but as compared to AD, in MF this was less. So this was actually, I think, a brief lecture, which I thought I'll share you the very recent studies which I have done in AD group, because we got really very interesting results. So what we concluded from this study is that chronic magnetic field stimulation in STZ rat model of AD, it has a significant effect on improving the cognitive deficits of spatial memory, recognition, and avoidance memory, and if given for longer duration. So mind you that we had given for 30 days and also for 60 days. So, and the amount of stimulation which we used was quite less. So this chronic magnetic field stimulation can improve cognitive deficits, and this improvement may be due to that there was maintenance of the synaptic plasticity or neuronal survival, or the amount of neurofibrillary tangles or the beta plaques which are found was significantly less as compared to the AD groups. So we propose that magnetic field stimulation could be a very effective therapeutic strategy in Alzheimer's disease, and if given for longer durations. So my acknowledgement to all the members, all the students of my lab of brain stimulation neuromodulation lab. So Abhishek is the one who did his PhD, and most of the work which I have presented is Abhishek. And then Reena is a postdoctoral student who has also done a lot of behavioral studies. Some of these behavioral studies which I have shown is done by Reena. And then there are other groups of members. I would like also to acknowledge ICMR for funding this as RA and SRF projects. Okay. Thank you all of you for patient hearing. I know this was a very small talk, but I thought to share with you the most recent findings from my group. Thank you. Thanks to all of you.
Thank you, Dr. Suman Jain, for this wonderful talk. We have some questions in the chat box. The first question is, what might be the mechanism of action after applying magnetic field in Alzheimer's disease? What might be the mechanism which is responsible for producing this therapeutic effect?
Yeah, so basically, I mean, if you talk of mechanism, what we are postulating that we haven't done any electrophysiological studies as of now. So this was basically, we initiated this study to see that starting from behavior to crude morphology, what is happening. Okay, we need to do more electrophysiology or go to the membrane level to see what exactly must be happening. But what is being given in literature is, like I said, that they're basically altering the membrane properties. Okay, so if you are altering the membrane properties, it has also been seen that they can alter the properties of especially the calcium channel. So maybe the calcium channel properties, if it is altered, then maybe a lot of transcription factor or activation of subsequent proteins may be getting, or the genes may be getting altered. So that could be one. Then it is also reported that if you give MF stimulation, then there is a release, the release of neurotransmitters is also altered. Okay. So that could be another mechanism by which it could be acting. So we need to do, I mean, more of these type of study to exactly see now. Now we know, okay, there is a effect on behavior and there is something, synaptic plasticity is being maintained. But actually now what is happening at the molecular level or at the channel level, then obviously we have to do more studies. However, in spinal cord injury, we had done studies. We did the calcium binding proteins expression, and then we had done some neurotransmitter studies also. So in those, we had seen that definitely the magnetic fields is altering the release of the neurotransmitters as well as the expression of the calcium binding protein. So till that, we have done some studies, but definitely we need to do. This is just a sort of, not really, I can say pilot study, because one PhD thesis is completed, but then this was a lot of work, but initiation of a mechanistic type of study. So that's what I can say.
Dr. Suman Jain, one question from my side. I wanted to ask you that how much time, what was the frequency of this magnetic field stimulation you will be giving to these rats so that they could able to produce this therapeutic benefit against memory impairment?
Yeah, so actually this is 50 hertz frequency. Okay, because what is basically in this chamber, magnetic field chamber, we can only alter the intensity, not the frequency, because that is how you are inducing the electric current, and it is a 50 hertz frequency. But this question is very pertinent because when we talk of application in human patients, then the coils which are there, basically we have to give either low frequencies or high frequencies. And there we are not talking of intensities. We generally talk of that if you're giving low frequency, then it is inhibitory. And if you're giving high frequency, that is excitatory. Okay. So that is why we now have purchased, so that we can have more application value. We have the rat coil which I had shown, and this rat coil will be now exactly similar to the human one. And so we will be doing studies, more molecular level studies using this coil, so that we can give low frequency and high frequency, which will be stimulating the condition as in done in human patients.
Okay. So Dr. Suman Jain, actually already what I know is that the transmagnetic stimulation is already carried out, or the clinical trials related to this transmagnetic stimulation is already carried out. And what I can see is that it doesn't penetrate too much deeper into the brain, and that's the limitation of this transmagnetic stimulation. So the diseases which generally affects cortex might get benefit more as compared to the diseases which are associated with the deep brain regions. So what do you think about the Parkinson's disease, where it is like the basal ganglia, which is quite deeper brain region? In that case, whether it is effective or not?
So yes, so that is another very, very important question. Yes, if you're using figure of 8 coil, then actually it doesn't go beyond 2.5 to 3 centimeters. Yeah. And the recent ones which has come, H coil, they say it can go up to 5 to 6 centimeters. Definitely that is there. But since lot of clinical trials has been conducted on Parkinson's disease, and there are reports which have shown that there is a recovery, or there is better improvement in the patients, which is also being observed for at least 6 months to more than that, 9 months also. Okay, so that is due to the stimulation of basal ganglia indirectly via the motor cortex. So that is how it is explained. So you cannot have direct stimulation of the basal ganglia. So it is an indirect effect which is causing these changes. However, now again in patients, there are some studies which have done single sessions or multiple sessions. So how many number of sessions you are giving, that will also decide that how much will be the changes. Because the mechanism basically in patients, or as we say, we are giving it repeatedly, because what we are trying to finally go into is to alter the synaptic plasticity. Okay, so if it is like in Parkinson's, we say it is a motor deficit, so therefore we give high frequency stimulations. Okay, and therefore, however, in say in schizophrenia or any psychiatric conditions, we give low frequencies. So the aim is to alter or modulate the synaptic plasticity, or maybe the release of neurotransmitter and other aspects of it, and that is how you are getting the effects. So what we have done, we recently, one of my MSc students, she is still doing. So rather than using RTMS per se, now we have a concept of theta burst stimulation, which is coming. ITBS, as we call it, intermittent theta burst stimulation or continuous theta burst stimulation. So she is giving a single session of intermittent theta burst stimulation. So this intermittent theta burst stimulation is excitatory in nature. So that single session, she has not done any follow-up study, but single session, she did find an improvement in UPDRS score. But that was done only for 1 day, and she is an MSc student, so obviously she couldn't do the follow-ups. But there is a change with even single session. And that has, for RTMS also, people earlier, they had shown that if you are given single trials or single sessions, then there is an effect.
Okay. Okay, thank you very much, Dr. Suman Jain. So I would like to thank all the speakers for today's
I think there was another
series.
question, if you don't mind, from
Yes.
Dr. Hemant Kumar. Can we increase the targeting for iron oxide nanoparticles in brain?
Yeah. That one, yeah. So
we can load
yes.
NOP for drug delivery in CNS.
Yeah, if you can load, like if you can target the iron oxide nanoparticles with this magnetic field, because if the drug is being released through this iron oxide, it may give some beneficial effects. So what do you want to say on this?
So yeah, so actually we did, I haven't shown in Parkinson's rat model, actually we implanted iron oxide nanoparticles in striatum, thinking that if you're giving magnetic fields, then maybe, local magnetic fields will be generated and maybe will have better effects. Because like you were asking that otherwise basal ganglia, the magnetic fields are not able to reach. So we may thought that maybe if you're implanting some iron oxide nanoparticles, maybe they'll get magnetized and will have magnetic effects local at the site of lesion. But then we did for 7 days, and then we did also for 1 month. But there was no cytotoxic effect, but there was no beneficial effect, and which could be due to, the brain is an enclosed structure, and iron, as we know, can have cytotoxic effect because of Fenton reaction also. So what we concluded from that study was that it could be that magnetic fields did not have any beneficial effects, but did not have any detrimental effects also. So iron as such, iron nanoparticles, it seems that in brain may not be very effective in conjunction with magnetic fields. So we are still on the lookout, and we are trying to develop magnetic field using some other particles, and we are in the process of doing that. If we can do that, then definitely this will be really good, and we'll be able to use it for drug delivery also, and maybe to, facilitate the effects of magnetic fields. Because we had actually done extensive work on spinal cord injury. So in spinal cord injury, actually what we did, we gave iron oxide nanoparticles into the lesion site of spinal cord, and then we gave magnetic fields. And you won't believe, it was marked effect. There was a lot of beneficial effect when we gave iron oxide nanoparticles along with magnetic fields. However, in brain that didn't work. So the reason what we are thinking is spinal cord is maybe more, open, and there was a lesion. I mean, there was a transaction in the spinal cord. So whereas brain is more enclosed structure with the less of, vasculature or the other aspects of it, that could have given differential effects. But from our studies, we know that the iron is not a good candidate for brain at least.
Okay.
In conjunction with magnetic fields.
Okay, thank you very much, Dr. Suman Jain. So now it was online, but we hope to see you in person at NIPER-Ahmedabad, to both of the speakers. So I would like to say thank you to Dr. Naren and Dr. Suman Jain for this wonderful talk and completing our second day. We will be having the sessions on the third day as well. So if you wish, you can join for that session. So thank you very much. On behalf of NIPER-Ahmedabad and Dr. Pallab, I would like to say thank you to both the speakers, and we would like to invite you in future in person here at NIPER-Ahmedabad. So thank you very much. A big round of applause for